Hello students, welcome to the EPG Pathshala. I am Dr. Imtiaz Hassan from Jamia Millia Islamia. Today we are going to discuss about a module tools used in bioinformatics under paper Bioinformatics. So the objective of this module is to train you about or you provide you the information about the different types of tools used in bioinformatics. Then in the tools, before using the tool we must know the basic information about the biological database. Then what are the classical tools in the bioinformatics that's most of the people used. Then we go for the tool one by one. For example, we discuss about the gene prediction tool, what are the tools used to predict a gene. Then we discuss about the sequence analysis tools, how to compare the sequences, how to analyze the sequences, how to derive information from the sequences. Then we discuss about the structure analysis tools, that how the function derived from the structure using the atomic coordinates of the molecule. And then at last we discuss about some miscellaneous tools that are used in the bioinformatics, how the tools are utilized actually. So the computational tools are routinely used for the characterization of gene, determination of a structure and physiological properties, then phylogenetic analysis, then performing simulation to study how the molecules are behaves in the living system. So for all the purpose, different types of tools are used. For example, if you want to know the physicochemical properties, the different sequences are analyzed based on the composition of the different amino acid, and on that basis we can derive a large number of information such as the molecular mass, ionic strength, molecular mass. Then we can know the hydrophobicity, hydrophilicity, then we can know the pi value, then bulkiness, then propensity for the secondary structure. So many information can be derived using the bioinformatics tool. Then the question arises that why, because of the vast amount of data there are generated, we must need a new approaches, and that approaches should be having the high throughput assay, and it's very difficult to manually access. That's why we need robotic system. Then speed of high computing added a very advancement in the biological system. That's why the biological research is running very fast. So to analyze these we must need the statistical information, and the bioinformatics actually help us to understand the large amount of data created through the biological research into a well organized system to understand all these information. We discuss about the different types of the database which is used as a tools in the bioinformatics. These database are the SwissProt, that's having information about the protein sequences, EMBL, that having information about the gene sequences, then the INTRACE, it's a collection of the tools, then GDB, it's contains the genome database, GenBank having the information about the gene sequences, GSDB, and the PDB, that's the containing information about the protein structure. We discuss about the classical tools in the bioinformatics. There are a large number of tools in the bioinformatics, but we gathered information about the very commonly used tools like the database interface for the GenBank, EMBL, DDBJ, Midline, SwissProt, PDB, etc. Then if you want to go for the sequence alignment, like if you want to see the alignment of the two sequences or more sequences, then you go for the BLAST, FASTA, Clustral, multi-align, D-align. These are the tools used for the sequence alignment. Similarly, if you have the protein sequence and you want to predict the structure, three-dimensional structure of the protein, that you can go for the Swiss modeler and the other modeling server. Then another tool for the gene finding. These tools are especially helps you to find out what are the sequence that's are of the gene, like the gene scan, genome scan, GenMark, GRAIL. These are the gene finding tools. The other groups of the tools that represent the protein domain analysis and identification. For example, if you have a protein sequence, you want to locate how many domains this protein have and where these domains are located for that, or you can find out from which family this protein belongs. That can be find on the tool PFAM, blocks, ProDom, or CDD. These are the tools used for the domain identification. Then the pattern identification or characterization, for that you can use the Gibbs sampler, Align ACE, or MEME. And in the last, if you want to do the protein folding analysis or folding prediction, that's used for the predict protein or the Swiss modeler. So these are some basic types of bioinformatical tools that can be utilized for the analysis of biological information. we provide you the idea that how actually tools used and what kind of tools can be used. You can see here that it's the genomic DNA, which is the after the transcription it's converted into the unprocessed RNA, which are after the processing this converts into the messenger RNA, and that messenger RNA is translated in by the ribosomes in the cytoplasm, and that leads to the protein polypeptide synthesize the nascent polypeptide chain, which converted into the folded three-dimensional structure protein of the and having the well specified three-dimensional structure that's necessary for the function. So the whole process from gene to protein involves a large number of steps, and for each step we can use bioinformatics tools to analyze. For example, at the sequence level we can use the tools for the ab initio gene prediction tools for the processing, and in this area we can use the comparative genomics tools, and finally for the protein we can use the tools for the functional identification. We can discuss there are two major gene prediction tools. One is the GRAIL that predicts the coding region in a protein. It uses a neuronal network which combines a series of codon prediction algorithm. It recognizing coding potential within a fixed size, especially the around 100 base windows, and evaluates coding potential without looking for the additional features. Later version incorporate additional information for the human and the other species. The another tool is the GenMark tool that based on the inhomogeneous Markov models and predicts coding region and non-coding regions based on the statistical pattern in the dinucleotide frequencies. We discussed the 5 very important websites that's used for that every biologist can use, and most of the biologists use the following websites. First is the NCBI, that's the mother of all bioinformatics server, where you can find information about almost every information about the gene, protein, and any biological information, including literature, compounds, and varieties of the information is available on the NCBI. Another is the EBI, that is the European Bioinformatics Institute, that also having the information about the genes and proteins. Moreover, it has varieties of tool for the gene and protein sequence analysis, and of course to gather the information in a very well organized format. Then the Canadian Bioinformatics Resources is also a well-known database, and it having the different types of the information, and is most widely used server. And the fourth is the SwissProt XPacy server. That's the SwissProt Bioinformatics Resources that having the information about, I think this website contains around more than 200 tools associated with this server. Apart from that, it having almost every information about a protein, from its function, from its activity, then association with the disease, then how the same gene is integrated or available on the different server. And the final fifth is the PDB, that's having the information about the protein structure. Protein structure is one of the most fundamental things in the biology, because the structure of a protein determines its function. So the PDB stores information about all the structure, all the proteins, their structure is known, and these structures are well annotated, and you can derive many information by using these websites. So these are the 5 websites which all biologists should know, and it's the most widely tools in the bioinformatics. We give you the basic information about the NCBI, that it having the interest interface to the database such as Midline, OMIM, then GenBank, gene peptide, then gene structure. The NCBI contains a BLAST server, which is the most widely used techniques or tools for the sequence finding or sequence comparison, or finding a sequence related to the particular genome sequences. Then it's having the draft of the human genome, as well as the many things are associated with the NCBI. We give the basic information about the EBI, which can be found on the URL www.ebi.ac.uk, and it's the SRS database interface, which is connected with the EMBL, SwissProt, and many more. And many tools are present on the EBI, such as the ClustalW, which is used for the sequence alignment, then DALI, which is used for the structure alignment and searching the similar structure comparison. It contains information about the pro function, where you can find the information about the function of a protein using their structure, and a large number of tools like interpro scan that help you to find the different types of motif located on this particular protein. And it is very user friendly, anybody can go and use it, and every information is written there, so you can directly understand what's the website want to convey the message. We discuss about the SwissProt. It is the present on the xpassi.org. It is actually the curated database, and having the extensive cross-linking with the other database. For example, if you have a protein sequences and you want to see the same sequence how it is available on the other website, for example, if you want to know the network, then it is directly associated you with the string database. So SwissProt is a gold standard by which other database can be measured, and it is the best place to start if you have any specific protein to investigate. So to do the sequence alignment, there are two ways to align the sequence. One, if you want to compare only two sequences, so for that, and if you want to analyze that what are the basic differences between two sequences, whether at the level of the mutation or at the level of the similarity. So these are the following are the there are many tools used for the pairwise alignment. One is the SIM, that's the used for the protein only, and it is the best known and interesting alignment in the xpassi server. Then Align software, that is optimal global alignment with no shortcuts. Then the L-Align, that calculates n-best local alignments. Then L-FASTA, it's the local similarity search showing the local alignments. Then BLAST 2, it's the local alignment using the BLAST at the NCBI, because the BLAST also give you output in the form of pairwise sequence alignment. And the last is the lab 2, which is the local DNA to protein alignment with the lab 2. So then we discuss about the multiple sequence alignment. The purpose of the multiple sequence alignment to see the conserved region in a particular family of the protein, particular types of the protein, to see the conserved sequences, conserved motif. For that, there are many tools are utilized. One is the well-known and most widely tool is the ClustalW. It's the global and progressive tool, and it is available on the EBI server. Then the CAP sequence assembly tool, that's used for the DNA sequence alignment. Then the MAP, it is used for the both DNA and protein. It's having the global progressive in a linear space. Then PIMA 1.4, that's used for the protein only. That's the actually protein pattern induced local multiple sequence alignment. Then MSA 2.1, that's used for the protein. That's the near optimal sum of the pair of the global. Then the block maker, that's used for the protein and finds conserved blocks in a sequence gained through the ClustalW. And the MEME 2.2, that's used for the both DNA, and it's mainly used for the elucidation of the motifs in a protein. We discuss about the BLAST. BLAST is the basic local alignment search tool, and it is there are different two types of BLAST mainly. BLAST P is BLAST of the protein sequences, and BLAST N is the BLAST of the nucleotide sequences. It's a nucleotide or protein sequences sent to the BLAST server, and it is compared against the summary of the matches. It is returned to the user. It allows combination of DNA or protein query sequences which searches against DNA or protein databases. So what actually it do? If you want to know that if you have any sequence and how many the proteins or the DNA sequences related to this protein, so you simply deposit that sequence, query sequences. It will give you the output in the format that it will give you all the sequences related to this protein or DNA, and what are the sequence similarity, what are the e-value, and what are the other outcomes. So you can easily compare pairwise alignment that each sequences comparison with the each sequence individually, and you can know the idea what are the region conserved between the two sequences, how they are similar, how they are differ in all these analysis. Especially it helps you to annotate the protein. If you don't know the function of protein, you can simply deposit the sequence to the BLAST. It will compare with the protein of the known function, and it will give you the basic idea about how this protein functions and what are the possible function of this particular protein. we give the information about the different types of blast, like for example blast P, which compares an amino acid query sequences against a protein sequences database. So it will especially use for the protein sequence analysis and it having the information about the protein sequence. Then blast N compares a nucleotide query sequences against a nucleotide sequence database. Then the blast X, it compares the 6-frame computational translation product of a nucleotide query sequences, both strands against a protein sequence database. T blast N, which compares a protein query sequence against a nucleotide sequence database dynamically translated in all 6 reading frames. And the T blast X, that compares the 6-frame translation of a nucleotide query sequence against the 6-frame translation of a nucleotide sequence database. So these are the different version of the blast. We discuss about the Prosite. It's the tool of the XPASI and it is maintained at the SwissProt Institute of Bioinformatics. All motifs are checked for false positive and fine-tuned. Sometimes a family can be defined by the more than one expression. It is the fingerprints and blocks automatically scans protein in a number of motifs. So the Prosite is mainly used to find out how many motifs are present in a protein sequence and where these motifs are located, how they are conserved, what are the probable functions. So all these analysis can be done at the Prosite website and it is associated with the XPASI server. We discuss about the PFAM. It is the protein family database. That PFAM7 contains a total information about 3,360 families and is a database of 2 parts. One is the curated and one are the automatically generated. It contains all hidden Markov models as a seed alignment which is added by using HMMER package. Now we discuss about the different websites. For example, in TMHMM, that's used to find out the prediction of the transmembrane helices present in a protein. So if you want to know that if a protein, whether it is a membrane protein or not, if a membrane protein, that it must have the transmembrane helices. So the propensity of a sequence to form a helix can be predicted by using hidden Markov model. So you can see here in a box, if you simply put your protein sequences and you can submit, it will give you the output that how many helices are present there and where it is the sequence are located. We discuss about the signal peptide prediction. If you want to know that whether a protein to be having signal peptide or not, so it can be predicted through using the SIGNALP tool. Like what is a signal peptide? Any protein has to be targeted to a specific part of the cell. It requires a signal peptide, and the signal peptide ensures that the protein is translated at the endoplasmic reticulum where it can enter to the secretory pathways, and that's the signal predicts that the cellular location other than the cytoplasm. So if you want to know that whether this protein is secretory or not, it can be predicted through the prediction of the signal peptide sequences, and that can be done by the using SIGNALP tool. We discuss about the protein structure prediction. If you have the protein structure sequences and its structure is not determined, you want to know what is the possible 3-dimensional structure of this protein, then there are various tools used for the protein structure prediction. Usually if you have the known protein with the good sequence similarity or good sequence coverage, then the homology modeling can be used. But if there is no good similarity, then we can go for the ab initio modeling, which is based on the energy maximization. Then the fold recognition method, that the sequence assigns the secondary structure, then align secondary structure with the corresponding secondary structure in a related protein sequences, and then accordingly it can give you the structure of the protein. And the third way is the statistical, that is based on the hidden patterns or the similar patterns or the similar structure. So these are the different models, different tools for the prediction of the protein structure. We discuss about the protein secondary structure prediction tool. For example, if you have a protein, every residue have their propensity to form the either alpha helix, beta strand or the coil, and of course their neighbor residue depends on the neighbor residue. So there are so many softwares are available to predict the secondary structure of the protein, like coil, that predicts the coil region in a sequence. For example, if you have sequence and you want to know where the coils are present, by using this tool you can predict how many coils are present. Then MN predict, it uses the two-layer neuronal network for the prediction of secondary structure. Then PSSP or SSP, that is the segment oriented prediction. Then PSSP or NNSSP, it is the nearest neighbor prediction. Then SAPS is the statistical analysis of protein sequences. Pair coil, it is the coiled coil region of the pairwise residue correlations. Then the protein hydrophobicity, hydrophilicity, and the SOPM, it is the self-optimized prediction method. So these are the various methods which can be utilized for the prediction of the secondary structure of a protein. We discuss about the structure analysis server. These are very commonly used server for the validation of the protein structure. For example, if you predicted a protein structure using any techniques, but it must be validated, otherwise the errors in the modeling cannot be observed or cannot be discovered. So to know that whether the structure is accurately modeled or not, the structure analysis verification server are used, and it's good that this server itself contains different components like the ProCheck that checks the stereochemical quality of a protein structure by analyzing residue by residue geometry and overall structure geometry derived from the subset of protein verification tools and from the what-if program. Then the WhatCheck, it does the extensive checking of the many stereochemical parameters of the protein model. It contains another tool, that is the ERAT, that analyzes the statistics of the non-bonding interaction between the different atoms and plots values of the error function versus position of the 9 residue sliding windows calculated by comparison with the statistics from the highly refined 3-dimensional structure. Then verified 3D, it determines the compatibility of an atomic model with its own amino acid sequences by assigning a 3D structure class based on the location of environment such as the alpha, beta, loop, polar, non-polar, etc. And finally, the proof, which treats the atoms like the hard sphere and calculates the statistical z-score deviation from the model from the highly resolved and the refined structure. So these are the different tools utilized for the structure validation on a server that's known as the SAPS structure analysis and verification server. We discuss about a very important tool, that is the analysis of a protein's 3D structure to help and identify its likely biochemical function. And this tool is known as the ProFunction. Is very user friendly. You can see here, you can just click here and you can upload the protein structure, and the structure are analyzed in many ways. Like example, it will compare with the other known structure to give you the information about the protein function. It compares the sequence also. It finds the motif using the Prosite, block, smart, PFAM. Then it goes for the fold analysis, like the fold and structure motif present, then the surface, clefts, residue connection. Then it also helps to find out the enzyme active site, ligand binding site, whether this protein have DNA binding site, yes or no. If yes, then where it is located. In that way, ProFunction give you a well organized structure analysis or the structure information of any protein that can be done on this server. In slide number 27, we discuss about the phylogeny reconstruction that constructs evolutionary trees based on the divergence that occurs in the related sequences. It is based on the parsimony or maximum parsimony or minimum distance. Then the parsimony construct tree to the number of mutation event is minimized. The two other tools are like the PhyLab, POP, and others, and some interactive tools. So these tools are used for the construction of phylogenetic tree and discuss about the evolutionary informations. We discuss about the protein interaction calculator. If you have a protein 3D structure and if you want to know how many interactions are present at the intermolecular level or intramolecular level, and especially if you have some ligand bind to the protein and you want to know how many interaction between the ligand and protein, you simply submit the PDB ID and it will give you, if it will analyze the structure and give you all the information about the context formed between the inside the protein atoms with the different atoms. Then that other tool is the protein fold similarity server. This server calculates the probability of identity between 3-dimensional domain. So it can use to perform the comparison of the submitted structure, and it will help you to compare with the other known structure of the protein. Then we discuss about the Foldex. It is a software which developed by the computer algorithm to provide the fast and quantitative estimation of importance of interaction contributing to the stability of the protein, like the interactions of the protein which measures the stability of the protein, and this gives you, based on the three-dimensional structure, you will get idea about the delta G value of the protein and how stable this protein. You can compare the stability of protein using simply the three-dimensional structure of a protein. We discuss about another very important tool. It is the P domains. It's a resource centered around the definition of assignment of structural domains in the protein domains. So it helps to find out the types of domains present in a protein. We discuss about another important server is the dial server, that's used to identify the domain identification algorithm. It is the server for the automatic identification of structural domains given in a three-dimensional structure of a protein, and the details you can find here. Then we discuss about the very important tool Globeplot2, that actually identifies the intrinsically disordered domain in a given protein structure. So in a protein, if you have a sequence, it is not necessary that all component of the protein is forming the proper 3D structure. So many structure are forming the intrinsically disordered protein. So you can locate that how many disordered region are present in a protein by using this tool, or you can simply submit the sequence and you get the information in the form of output. Another structure analysis tool, and it is a web suite for the protein structure analysis. If you have a protein structure and you want to analyze in many ways, you can analyze in a single server. Like for example, if you want to see the Ramachandran plot, if you want to analyze the temperature factor, if you want to know the intermolecular interaction, if you want to know the interaction of a particular residue, display of the interfacial residue, if you want to see where the disulfide bond are located and how it is located, all these analysis can be done on a single server. There's the PSAP, that is the protein structure analysis. You know that in the bioinformatics tools is used to analyze both the genomic data as well as the proteomic data. So now we discuss how in the proteomics the bioinformatics helps, and how the bioinformatics fasten the process of proteomics. Because for that we first know what is the proteomics. Proteomics is the cataloging and analysis of proteomes, or the complete set of expressed protein in a cell at a given time. And proteomics focuses on which protein are made and what quantity, and their interaction with the other. So in a proteomics data you got many things. First, that what kind of protein expressed in what condition, then what are the relative concentration of what are the relative level of expression of these proteins, and do these protein having any interaction. So the goal of proteomics is to identify every protein in the proteome using 2D gel electrophoresis, mapping them, then isolating each protein and analyzing it by mass spectrometry to identify what are actually these proteins. And then bioinformatics comes, and that develop a database with a sequence of each protein and analyze the protein at the different cell type and stages of the development. So in the proteomics, the bioinformatics actually help us to analyze the proteomic information, that you can compare the 2D gels of the two samples, that how these 2D gels are differ, do any relative expression of particular protein changes or not, if changes then at what concentration. Then we can correlate the 2D page data with the level of disease, association with the disease, then the progression of the disease, prognosis of the disease. So the bioinformatics actually helps to analyze the data in a very, very fast way. Otherwise it's very hard to analyze the proteomics data. Then the protein identification and sequencing is a very complex process, especially the cellular genomics is involved in the identification, sequencing of and computer analysis of the data. Proteomics stands to make a major contribution to understanding the human diseases and development of biopharmaceutically based diagnosis and treatment. So actually proteomics helps to identify the molecule, because if you get the sequencing data, it's very hard to analyze one manually. But in a bioinformatics, if you use the bioinformatics tool, you just simply submit the sequence and you will get a lot of information by analysis of these sequences using the integrated database, and then it will give you all the information in a well precise manner it's very hard to determine manually. That's why the inclusion of bioinformatics to the proteomics actually made this field very, very innovative and very useful for the biological research. So I hope you understand very well that what are the different tools, how these are utilized. We can summarize that the bioinformatics is a comparatively young discipline but have progressed very fast in the last few years because of the advancements in the high-speed computing facility, and it has made it possible to test our hypothesis virtually and therefore allow to take better and informed decision before launching costly experiment. For example, if you want to design a drug, so for that you have to synthesize the chemical, go for the binding study, for that you need protein, then again you go for all the evaluation, and at the end you find nothing happened. So that's why to reduce such costs, you first do all the analysis in silico, that what kind of molecule will preferably bind to what kind of protein, then you select that protein, what is the binding affinity, all the things can be predicted through the bioinformatics, and then you can design the experiment to perform. So it will reduce your cost, it will reduce your time, and it will reduce your energy. So to compare the more than two sequences, multiple sequence alignment can be performed, which is based on the fact that the similar sequences are usually homolog, because if you take the two sequence and if you want to compare manually, it seems to be impossible to compare manually. But if you do the multiple sequence alignment, within a single click you get all the information that how these two sequences are related, what are the differences, what are the similarity, where the sequences are similar, where are the sequences are different. So with the evident advancement in the computational hardware and software, most of the drug discovery steps are performed in silico, and then the experiments are designed further. And the several software are available to perform homology modeling to get the fast structural solution of the given protein sequence, because to determine the structure you either go through the NMR or go through the crystallography. It's a very tedious job. It needs a longer time, long energy, longer time to grow the crystal in the crystallography. In the NMR it takes a longer time, but in a homology modeling you just go submit the sequence and it will give you the well evaluated model for the three-dimensional structure of a protein. That's why bioinformatics helped the recent biological field, and the application of bioinformatics tool helps to get the large number of information derived from the biological data sets.